Greetings, Brookings Bio students. This is Mrs. Rydell, and in this slideshow, we're going to take a look at what happens to those pyruvic acids if there is oxygen available to the cell. If you remember from our last slideshow, we talked about glycolysis, splitting our sweet. We're going to take a glucose, sugar molecule, and break it in half into two pyruvic acids. And then the cell has a choice about where it's going to go next. And the choice is determined by whether or not there's oxygen available, enough oxygen available to the cell. If there's not enough oxygen, we're going to follow the fermentation pathway, either lactic acid or alcoholic. And we talked about that in our last slideshow. In this slideshow, we're going to look at what happens after glycolysis if there is oxygen available. And the cell is going to follow a different path. It's going to enter into what we call the Krebs cycle and then onto electron transport chain. So let's take a look at a little video that's going to kind of walk us through the next steps.
All cells need energy to stay alive. Usually your cells get their energy through the process called aerobic respiration. The word aerobic refers to oxygen. In aerobic respiration, oxygen interacts chemically with a molecule of glucose. The glucose is broken down into carbon dioxide and water. This process releases energy, which the cell stores in the chemical bonds of ATP. The first stage of aerobic respiration, glycolysis, occurs in the cytosol, the jelly-like part of the cytoplasm. Glycolysis breaks each molecule of glucose into two molecules of pyruvate, energizes two electron carrier molecules, and produces two molecules of ATP. The next two stages of aerobic respiration are carried out in the mitochondria. Pyruvate enters a mitochondrion. Once inside, it is modified and enters the Krebs cycle. The reactions of the Krebs cycle break the pyruvate molecules down to carbon dioxide, create two more molecules of ATP, and energize more electron carrier molecules. The final stage of aerobic respiration is electron transport. The electron carrier molecules that were energized in the earlier stages deliver electrons and hydrogen ions to the electron transport chain. As the electrons are passed down the chain, energy is released and used to make ATP. This final stage produces as many as 32 molecules of ATP from each molecule of glucose.
Okay, so let's look at the steps the cell is going to follow now. Remember, we're starting with glucose, C6H12O6, and this process is the with oxygen path, so we're going to use oxygen, O2. And remember, glycolysis can happen with or without oxygen. So we're going to go through glycolysis like we did in our last slideshow, split our sweet. We're going to break our glucose into two pyruvic acid molecules, also called pyruvate. And then we're going to send them into the Krebs cycle. Remember when we talked about photosynthesis and we had the Calvin cycle named after Melvin Calvin? This cycle is also named for a person. His last name was Krebs, Hans Krebs. And so we're going to see the steps now as those pyruvic acids, those pyruvates, enter the Krebs cycle and follow the next part of the pathway. So if you look at it, it's kind of like remember we did photosynthesis. There's a lot of in-betweens. There's kind of some chemistry going on here. And we're not going to learn all the intermediates, but we're going to kind of do what we did with photosynthesis. What's coming in and what's coming out. And this pathway now, the Krebs cycle, is the next step that the cell is going to follow if oxygen is available to the cell. So we're going to bring in our pyruvic acids here, and we're going to charge them up, get them ready, and we're going to feed them into the Krebs cycle. As it goes through the cycle, we're breaking apart those carbon molecules little bit at a time, just like we talked about, little baby steps. And when we're doing that, we're releasing, charging up some electron carriers here. We see that NADH and FADH2. We're going to make a little ATP, and we're going to make some carbon dioxide. That's a waste gas that organisms doing cellular respiration give off as they exhale when we talked about respiration. So let's look a little closer at the steps here. We're going to watch a little video and see what happens in the Krebs cycle.
The 3-carbon pyruvate molecules formed during glycolysis still contain most of their chemical energy. This energy is harvested in a series of reactions called aerobic respiration. Aerobic respiration takes place inside the organelles called mitochondria. The pyruvate molecule passes through the outer membrane of the mitochondrion and into the outer compartment. The pyruvate molecule then passes through the inner membrane and enters the mitochondrial matrix. The first series of chemical reactions in aerobic respiration takes place in the mitochondrial matrix. The pyruvate molecule is split and enters a continuous loop of reactions. This loop of reactions is called the Krebs cycle. As these reactions take place, energy from the carbon compounds is used to make NADH, ATP, and FADH2, all high-energy compounds. Carbon dioxide also forms and is released. These carbon dioxide molecules leave your body when you exhale. Before entering the Krebs cycle, another pyruvate molecule is split. More carbon dioxide is released. Next, 2 electrons are transferred from the 2-carbon compound to NAD, forming NADH. The 2-carbon compound becomes attached to a molecule called coenzyme A. This forms the highly reactive molecule acetyl-CoA. Acetyl-CoA enters the Krebs cycle, and the acetyl portion becomes linked with a 4-carbon compound. The result is a 6-carbon molecule called citric acid, the first intermediate in the Krebs cycle. 1 carbon is removed, taking with it 2 oxygen atoms. The resulting molecule, carbon dioxide, is released. The 5-carbon molecule continues through the cycle. 2 electrons are transferred to an NAD molecule. NADH is formed. A similar sequence of events takes place again. This time, the 5-carbon compound is changed to a 4-carbon compound. Another carbon dioxide forms and exits. Another pair of electrons is transferred to NAD, producing NADH. The leftover 4-carbon compound is still energized. It gives up a bit of its energy to an ADP molecule, forming ATP. Next, a pair of electrons is transferred to a different electron carrier called FAD. This forms higher energy FADH2. In a new reaction, a slightly different 4-carbon compound is formed. One last reaction of the Krebs cycle liberates 2 more electrons from the 4-carbon compound and converts another NAD to NADH. The remaining de-energized 4-carbon compound is the starting material for a new round of the Krebs cycle. Recall that a glucose molecule is split into 2 3-carbon pyruvate molecules during glycolysis. Therefore, the chemical reactions in the Krebs cycle occur twice for each glucose molecule that enters the glycolysis pathway. This produces a substantial harvest of high-energy compounds. These high-energy compounds remain in the mitochondrion and power the final phase of aerobic respiration, the electron transport chain.
So let's kind of review a little bit about where we came from and how we got here. Glycolysis is going to happen in the cytoplasm outside the mitochondria before it even gets into our power plant. We're going to make those pyruvic acids by splitting our sweet. The Krebs cycle is going to happen in the matrix inside the mitochondria. So once we do glycolysis and make our pyruvic acids, we're going to send them into the mitochondria way inside here. Remember, the matrix is in the middle here, in the center of the cristae. And that's going to be the place where our Krebs cycle is going to happen. So we're going to take those pyruvic acids, also called pyruvate, that we made during glycolysis, and we're going to send them into the mitochondria, and they go through the outer membrane, they go through the cristae membrane, way into the matrix, and they're going to join with a special helper molecule called coenzyme A. Coenzymes are molecules that help enzymes work, and this is called coenzyme A. It's made from vitamin A. That's one of the reasons that you need to eat a balanced diet and get your vitamins in your food, is that a lot of those vitamins act as enzyme helpers to help your chemistry happen inside of you. And this is coming from vitamin A in your food, and it's made into an enzyme helper, and that enzyme helper is going to join with pyruvic acid and turn it into a form that is going to be able to enter the Krebs cycle. And it makes a molecule called acetyl-CoA. So here's what we're going to do. We're going to join coenzyme A with a pyruvic acid. In order to do that, we're going to remove one of the carbons here. And whenever you break a bond and release carbon, oftentimes it's going to go off as a gas molecule, as carbon dioxide gas. So we're going to send one of those carbons out as a gas, as carbon dioxide, and the remaining 2 carbons are going to attach to coenzyme A. Here they are here, 2 carbons from pyruvic acid attached to that coenzyme A molecule to make this combination molecule, acetyl-CoA. It's part carbons from pyruvic acid, part coenzyme A. So when that happens now, the carbons that we remove are going to be released as carbon dioxide into the atmosphere. And we're going to charge up now. When we break a bond, we can charge up some of those high energy electron carriers. The NADH that we make when we split off that carbon to make carbon dioxide and feed those carbons from pyruvic acid into the Krebs cycle are going to move on to the next part of our pathway, the electron transport chain. We're going to come back to that after we talk a little bit more about the Krebs cycle. So we're going to take now our acetyl-CoA, coenzyme A, with the 2 carbons attached to it, and we're going to feed it into the Krebs cycle by hooking these 2 together with a 4-carbon molecule to make a molecule that's got 6 carbons. 4 plus 2 is 6. It makes a molecule called citric acid. Remember I said we don't have to know all of the pathway and all of the intermediates of the cycle, just like before when we did Calvin's cycle. But we are going to learn this one because this is a pretty important one. It's the first one that forms when those carbons from pyruvic acid feed into the cycle. And another name for the Krebs cycle is the citric acid cycle, and that's because this is the first molecule that gets formed when we combine those carbons from pyruvic acid into the Krebs cycle. So we're going to watch a little animation about how this works. Okay, so follow now. Here's my edge of my mitochondrion. Here's my cristae, my inside membrane. Here's my outside membrane. Remember, out here we're in the cytoplasm, outside the mitochondria. I'm going to take my pyruvate now, my 3-carbon molecule I made during glycolysis, and I'm going to pass it into the mitochondria. There's transporters that do that. I'm going to attach it to CoA. Let's try that again. Feed it in, attach it to CoA by removing one of the carbons. It goes out as carbon dioxide. And then now, here's my 4-carbon molecule from the Krebs cycle. I'm going to feed those 2 carbons into the Krebs cycle. And then as it's passing, it's going to move through the cycle. We don't have to know all the intermediates, but watch what's coming out. The carbon dioxide comes out, NADH comes out. We're going to make some ATP, a little bit of ATP. We're going to charge up some FADH2. We're not worrying about the intermediates, remember. We're worrying about what's coming in and what's coming out. Notice those carbons that got fed in from pyruvic acid all ended up as part of a carbon dioxide molecule that was released out to the atmosphere. You exhale carbon dioxide when you breathe. So let's follow this again. Pyruvic acid in. We're going to charge it up with CoA. And then we're going to feed it into the Krebs cycle. We make citric acid first. And then running through the cycle, watch what's coming out now. We're going to charge up some electron carriers, NADH. We're going to release carbon dioxide. release a little ATP, release some FADH2. Notice what happens to our CoA now. It recycles. It's a helper molecule, and it's ready to receive the next pyruvic acids coming in and feed them into the Krebs cycle. So they bring it in, drop it off, and get recycled again. So coenzyme A goes back. It's going to pick up another pyruvic acid, bring it into the cycle, drop it off, go back and pick up another pyruvic acid, bring it into the Krebs cycle, go back. And then the citric acid that's formed when those carbons from pyruvic acid join the Krebs cycle is going to go on to complete the rest of the cycle. Remember, we said don't worry about the intermediates now. What's coming in? Pyruvic acid is going to feed into the cycle. What's coming out now is carbon dioxide. From these 3 carbons in pyruvic acid, we're going to get 3 carbon dioxide molecules. We're going to get 1 ATP. We're going to get 1 FADH2. And we're going to get 4 NADH. We're charging up electron carriers. We're charging up a little ATP. And remember, we want to break down the glucose molecule a little at a time, baby steps. And that's what we're doing. We're not releasing the power all at once. We're taking off the carbons one at a time and slowly releasing the power so we can capture it in these electron carriers and capture it in ATP. Every carbon that comes in, it's going to go out as a carbon dioxide, and as we break those bonds, we're saving the power in these high energy carriers. The glucose, the carbons that started in glucose, are going to go out as carbon dioxide. They're lost to carbon dioxide into the atmosphere. Remember, when you breathe, you're taking in oxygen, using it to do cellular respiration, and then exhaling carbon dioxide gas as a waste product coming from burning our sugar. The ATP that I made during the Krebs cycle can be used directly by the cell. It's a power molecule. ATP can be used for transport, can be used to do chemistry, all those things that we talked about that we need power for, growth, repair, reproduction, all of those things need power, and that ATP that we're making directly from the Krebs cycle can go right into the cell and be used to provide power for something. These high energy electron carriers, the NAD and FAD, NADH and FADH2, are going to move on to the next part of the pathway, which is the electron transport chain. So on our next video, we're going to focus on electron transport. What happens next? Stay tuned.